Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
Welcome to the debate. So, imagine inventing a miracle organism, right? Like a microbe that could cure a rare disease, or maybe completely replace petroleum-based plastics.
Which sounds amazing until you realize the logistics.
Exactly, because then you realize that keeping that organism alive in, a massive 1,000-liter steel tank is going to bankrupt your company before you sell a single drop.
Yeah, it's a brutal reality. I mean, the cell is the factory, and the factory has a mind of its own. It reacts to its physical environment in totally unpredictable ways. It's basically the ultimate hostile manufacturing environment.
Right, and managing that unpredictability while actually surviving as a business, well, that's the central challenge for the fastest-growing sector of our industry. So today, we are unpacking a core subject drawn from the 2025-2026 Industry Review.
Specifically looking at collaborations between small biotechnology companies and contract research organizations, or CROs, in microbial fermentation.
Yeah, and just to define our terms, by small biotechs, we mean companies with under 50 employees and under $50 million in revenue.
It's such a fascinating dynamic, honestly, because it's defined by this massive paradox. Small biotechs are the indisputable engines of innovation right now. I mean, they account for 22% of all EU biotech patents.
Wow, 22%?
Yeah. They are pioneering precision fermentation for alternative proteins, recombinant enzymes, sustainable chemicals. They have the brilliant ideas, and they have the genetic engineering chops.
But they face just brutal physical constraints. To build a standard 10,000 square foot pilot plant in-house, that costs between $6 and $10 million in upfront capital.
Which they don't have.
Exactly. And it takes 18 to 36 months of lead time just for the building, the stainless steel piping, the equipment, and the validation. Startups simply do not have that kind of time or money to burn.
Right.
As a result, the latest BioPlan survey shows that microbial outsourcing by these small sponsors has now crossed the 53.7% threshold.
Which brings us to the actual core thesis of the 2025-2026 review, which strongly recommends what it calls the hybrid model.
The hybrid model?
Right.
This is the practice of highly structured outsourcing of fermentation optimization and scale-up, while the small biotech keeps the core strain engineering in-house. Basically, they design the organism and someone else scales it up. And the specific question we are debating today is this hybrid model a bulletproof blueprint for small biotechs, or does artificially separating the strain development from the scale-up introduce, systemic vulnerabilities that complex contracts just can't fix?
That's the crux of it.
I represent the position that the data is definitive here. The structured hybrid outsourcing model, governed by rigorous scorecards and milestone contracts, is the unequivocally optimal strategy that neutralizes capital deficits and actively accelerates timelines.
Okay. And I represent the position that while this hybrid model is, super financially appealing on a spreadsheet, bifurcating biological development at early stages introduces profound operational and technical fragilities that standardized scorecards just mask rather than actually solve.
Well, let me lay out the foundation for why the hybrid model is not just a successful option, but a strictly necessary evolution for the industry. The economic realities of bringing a biological product to market are staggering.
Oh, they're terrifying.
Terrifying is the word. We are looking at process development costs of $60 to $190 million per market success when you adjust for inflation and clinical attrition. A small biotech literally cannot absorb that. By outsourcing to specialized microbial CROs, small companies achieve $4 to $8 million in direct capital avoidance per project.
Sure, on paper.
Not just on paper, in reality. They don't have to build dedicated fermenters, utilities, or clean rooms. Furthermore, they cut their timelines drastically. Instead of taking 18 to 24 months to optimize a process in-house, these specialized CROs are doing it in 9 to 14 months.
But with what yield?
Let me finish this point. With structured milestone contracts and design of experiments protocols, the tech transfer success rates are hitting an incredible 85 to 95%. I see why you think that separating these functions introduces fragility, but let me give you a different perspective. This isn't a loss of control for the small biotech. It is an immediate strategic upgrade to specialized capabilities they could never, ever afford to build themselves.
I'm sorry, but I just don't buy that. Let me tell you why. The review portrays this hybrid approach as a seamless plug-and-play model. And frankly, that is a dangerously optimistic view of biology. Biology intensely resists clean corporate handoffs.
Come on, it's not that dramatic.
It is. The hybrid model recommends handing over a biological process at technology readiness level 3 or 4. That is essentially a proof of concept in a tiny lab flask or maybe a 2-liter benchtop bioreactor.
Which is standard.
But doing so creates a massive, perilous gap between the biological scientists who engineer the strain and the chemical engineers at the CRO who are trying to scale it to 1,000 liters. And those 85 to 95% success rates you just quoted?
From the data, yes.
They do not happen by magic. Achieving those metrics requires immense, often completely hidden costs. The review's own best practices literally require allocating 10 to 15% of the entire project budget exclusively for joint oversight and communication.
So, communication is good.
It proves my point. It proves that the risk is not fully mitigated by the CRO's infrastructure. The friction is inherent to the physical separation itself.
Okay, but if the hybrid model is saving these startups up to $8 million right out of the gate, we have to ask what they are trading away to get that discount. And I argue they aren't trading away anything. They are tapping into a vastly superior paradigm.
A superior paradigm?
Yes. Let me offer an analogy to frame the economics here. Outsourcing pilot plants in modern biotech is exactly like a software tech startup utilizing cloud computing instead of building their own physical server farm.
Oh, I don't like where this is going.
Just hear me out. 10 or 15 years ago, a tech startup had to, buy servers, rack them, cool them, maintain them. Today, they rent AWS or Azure. Why on earth would a 30-person biotech company spend $10 million and wait 3 years to build a GMP facility when boutique microbial CROs provide immediate, on-demand access to platforms ranging from 1 liter all the way up to 10,000 liters?
Right, but
By plugging into that shared infrastructure, small biotechs reduce their overall development costs by 20 to 35%. It is literally the democratization of biomanufacturing.
I have to reject that analogy entirely. I really do.
Why? It maps perfectly.
It doesn't, because cloud computing works because code is static. A Python script runs exactly the same way on a server in Virginia as it does on a server in Tokyo. Biological organisms are not static code.
I know they aren't code, but
They are highly dynamic living systems that react acutely to their physical environment. When you take an E. coli or a Pichia pastoris yeast strain out of a 2-liter benchtop reactor and put it into a 1,000-liter stirred tank, the physics change dramatically.
wait, let me stop you there just so we are perfectly clear on the mechanics for a second. Are you saying the actual physical pressure in the larger tank actively alters the organism's biological behavior? How does a bigger tank change the biology?
Absolutely, it alters it. Fundamentally, think about shear stress from the impellers, the massive blades that mix the fluid in the tank.
Okay.
Force required to mix that much liquid turns those impeller blades into something resembling an industrial blender.
An industrial blender, that's a bit hyperbolic.
It physically tears the cells apart, or it stresses them so much that they literally stop producing your target protein. And then there is the oxygen transfer rate, or KLA. In a small flask, oxygen diffuses super easily. In a massive tank, you have literal dead zones where the cells suffocate and zones where they are over-oxygenated.
Right, but
You cannot just upload a cell line to a CRO and expect it to execute like software. Generalist clinical CROs frequently underperform in high cell density fed batch control precisely because of this.
So wait, you're telling me that a team can spend 2 years engineering a perfect cell, and the moment it goes into a bigger tank, it essentially forgets how to produce the protein because it's too stressed out by the environment?
Exactly. And if that physical environment isn't matched perfectly by the CRO, the financial savings you are championing vanish instantly. The review itself cites an example of an ad hoc transfer that resulted in a 35% drop in titer, meaning the actual amount of product the cells produce, and a 6-month delay. That is the reality of biological transfer.
But that 35% titer drop in 6-month delay was specifically from an ad hoc transfer, which is exactly what the structured hybrid model is designed to eliminate. You bring up the changing physics of the bioreactor, right? Oxygen transfer, shear stress, dead zones.
Yeah, the things that kill the cells.
Executed with a specialized microbial CRO is so superior. These boutique CROs have the computational fluid dynamics, the CFD-validated scale-down models to match that volumetric mass transfer coefficient before they ever move the organism to 1,000 liters. They basically simulate the industrial blender in the gentle whirlpool.
Well, simulating it and surviving it are two very different things.
But let's look at the actual performance data from the review regarding that TRL-34 handoff you mentioned earlier. When a CRO takes over at that benchtop proof of concept stage, the starting titers from the small biotech labs are usually sitting around 1 to 3 grams per liter.
Sure.
Within 6 to 9 months, the CRO routinely drives that titer up to 5 to 15 grams per liter. That is a 2x to 5x improvement in productivity.
Okay, and how exactly are they achieving that massive leap so quickly when the originators, the people who literally built the strain, couldn't?
Through infrastructure the small biotech could never afford. They apply parallel design of experiments, or DOE, in high-throughput microbioreactors like the Amber 250 system.
Ah, the Amber 250.
Yes, because if you're thinking, well, why don't the startups just test different nutrient feeds themselves? Testing one variable at a time takes years. The Amber 250 is an automated system that runs 24 tiny, fully functional bioreactors simultaneously. A robotic arm feeds them, adjusts pH, and tweaks oxygen levels autonomously based on an AI-driven matrix of thousands of variables.
It's a nice piece of tech.
It is. Small biotechs simply do not have the capital to run 24 automated microbioreactors simultaneously to test media, feed strategy, and induction profiles. Specialized CROs are fundamentally better equipped for process optimization than the scientist who invented the strain.
That's a compelling argument. But have you considered the integration of downstream processing, or DSP? You are entirely focused on the upstream, the fermenter, the titer, the robotic arm tweaking the oxygen.
Well, upstream is where the product is made.
Yes, but getting 15 grams per liter of product inside the fermenter is utterly useless if you cannot purify it efficiently afterward. This is where the strict hybrid separation at TRL-34 becomes incredibly dangerous.
How so?
When you isolate strain engineering from the realities of large-scale downstream purification, you build in catastrophic inefficiencies.
Walk us through that then. Why does high upstream yield guarantee a downstream problem?
Because the cell is a complex package, and eventually you have to break it open and clean up the mess. If DSP isn't integrated from day one, you routinely face 20 to 40% yield losses right at the interface between harvest and purification. Let's take the review's benchmark on host flexibility as an example.
Okay.
A small biotech might spend 2 years engineering an E. coli strain to produce a recombinant enzyme. Now, E. coli is great for upstream yield, but E. coli naturally produces endotoxins in its cell wall as a byproduct of simply existing.
Right, which have to be removed.
Exactly. If you use it, the CRO has to spend an absolute fortune and add multiple complex filtration steps to wash those toxins out later. Otherwise, the product is literally unsafe for humans. Sometimes, to make the downstream process viable and reduce endotoxin removal steps by 50 to 80%, you have to completely abandon E. coli.
Then switch to what?
You switch the host organism entirely to a yeast like Saccharomyces or Pichia, which just don't produce those endotoxins in the first place.
So the strain engineers optimize the wrong organism because they aren't thinking about the purification at the end of the line.
Precisely. If the strain engineers are operating in a silo at their 30-person startup, isolated from the CRO's scale-up and downstream engineers, they might spend a year optimizing an E. coli strain that is fundamentally unpurifiable at commercial scale. Strain and process are symbiotic. They are not sequential steps on an assembly line that you can just toss over the fence when you finish your part.
I entirely agree that strain and process are symbiotic, which is exactly why the structured governance model in the 2025 review mandates early integration. You don't just toss the E. coli over the fence and wish them luck.
But that's what happens in practice.
No, the best practices dictate conducting a joint techno-economic analysis, a TEA, within the very first 4 weeks of the partnership.
A TEA is just a financial projection. It's a spreadsheet.
It is much more than that. That TEA sets quantitative targets for tighter, yield, productivity, and cost of goods sold before any heavy optimization begins. It forces the startup and the CRO to sit in a room and say, wait, if we use E. coli, the endotoxin cleanup will ruin our profit margins. We need to switch to yeast right now.
Assuming they catch it.
It actively prevents those downstream bottlenecks you just mentioned. Look, if we accept that biology is messy, how do we control the mess? We engineer frameworks. Look at the 100-point weighted scorecard for CRO selection outlined in the review.
I have deep reservations about those scorecards.
But the data supports them. When small companies use this scorecard, specifically making sure they weight microbial fermentation expertise at 25%, they achieve a 92% on-time delivery rate.
92%.
Yes. They are systematically evaluating CROs on having 5 validated microbial hosts, commercial-scale references, and digital twin capabilities. And on top of that, you have a 30-40-30 milestone payment structure.
Let's talk about that milestone structure.
30% up front, 40% only upon hitting key deliverables like that 5 gram per liter titer, and 30% on the final tech transfer package. There are even clauses where the CRO absorbs 10 to 20% of the scale-up failure costs. This contractual framework practically eliminates the sponsor's financial risk while solving the biological integration problem.
Practically eliminates? A 10 to 20% risk share absorption by a CRO is cold comfort to a small biotech whose single foundational asset is delayed by a year.
It's better than eating 100% of the cost.
If you are a 30-person startup burning $2 million a month and your pilot run fails because the cells sheared in the massive impellers, the CRO giving you back a fraction of the milestone payment doesn't save your company from bankruptcy.
Well,
you brought up the cloud computing analogy earlier, and I actually think it applies perfectly here, just not how you intended. A scorecard is just a service-level agreement for a server, but you cannot sign an SLA with a living cell.
But the SLA isn't with the cell, it's with the engineers managing the cell.
And let's look closely at what it actually takes to make those engineers deliver on those contracts. The so-called best practices you are citing require weekly joint steering committee calls. They require implementing digital twins.
Which are incredible tools.
Just to clarify for anyone wondering what a digital twin is, it's essentially trying to build a perfect virtual simulation of the 1,000-liter tank on a computer to predict how the cells will behave. And they have to do that just to reduce batch-to-batch variability to under 5%.
Exactly. It's sophisticated engineering.
The guidelines also require mandatory co-location, where the biotech scientists have to physically fly out and literally live at the CRO facility for 1 to 2 weeks during the transfer.
That's just good teamwork.
The sheer volume of mitigation strategies required. The shared electronic lab notebooks, the digital twins, the 15% budget allocation just for oversight, it proves my point perfectly. The friction of outsourcing biology is massive. You can draft the most elegant 100-point scorecard in the world, and even if a top performer scores a 95, that piece of paper cannot replace the intrinsic, institutional knowledge of a team that engineered the organism from day one and scaled it in the very same building.
But building that building takes 3 years and $10 million they do not have. The institutional knowledge argument is, frankly, romantic, but it completely ignores the brutal reality of capital markets in 2026.
I'm not being romantic. I'm being practical about the biology.
These aren't evidence of a broken model. They are evidence of a maturing, highly sophisticated model. Yes, there is biological friction. No one is denying that moving a living organism from a 2-liter flask to a 10,000-liter stainless steel tank requires intense coordination. But the structured framework works.
Sometimes.
Look at the precision fermentation enzyme case study from the review. A small startup was stuck at a titer of 1.8 grams per liter in-house. By partnering with a microbial CDMO, utilizing their DOE and high cell density fed batch expertise.
And fed batch is notoriously difficult to master.
Exactly. because fed batch means continually feeding the cells nutrients while they grow, rather than giving it all to them at the start. It sounds simple, but what happens if you feed them too fast?
You trigger overflow metabolism. When you feed cells too much glucose too fast, instead of making your target protein, they essentially panic and produce acetate or ethanol. It turns the tank toxic. Managing that feed rate in a 1,000-liter tank where mixing isn't instantaneous is incredibly difficult.
Which is exactly why the startup needed the CRO. They couldn't manage the overflow metabolism in-house. The CRO stepped in, managed the feed strategy, and hit 7.2 grams per liter in just 9 months.
Wow.
That is a 4x improvement. Their overall recovery was 78%, and they reduced their cost of goods sold by 35%. More importantly, they reached 100 liters of GMP material in 14 months, whereas doing it in-house would have taken them 26 months. That 12-month acceleration, that's the difference between securing Series B funding and going out of business entirely. The biological friction is manageable. The capital deficit of doing it in-house is fatal.
All right, I will concede that the capital constraints are an intractable reality. Small biotechs cannot just magic $10 million out of thin air to build GMP pilot plants.
Exactly.
And to that end, outsourcing to specialized boutique microbial providers rather than large generalist clinical CROs is an absolute necessity. The generalists simply lack the nuanced capability to manage that overflow metabolism we just discussed.
Right.
But the industry must stop pretending that the hybrid model is fundamentally frictionless just because we have better contracts and AI robots now. Biology is inherently messy. The fact that top-tier CROs must offer 5 to 10 validated hosts, and the fact that we frequently have to switch from E. coli to yeast midstream just to make purification possible shows that scale-up is often a process of biological triage, not predictable engineering.
It's managed triage.
When a company separates the scientist who designed the genetic circuitry from the engineer who manages the oxygen transfer rate, they are building a vulnerability into their product's DNA. Contractual milestones don't solve oxygen shear stress. Scorecards don't optimize harvest yields. The intimacy between the strain and the process is an absolute biological necessity. And an outsourced contract will always, fundamentally, struggle to replicate the efficiency of a single integrated team.
Yet the data demonstrates that when that intimacy is simulated through rigorous frameworks, like doing the joint techno-economic analysis within the first 4 weeks, setting up shared digital infrastructure, and leveraging those AI-enabled DOE platforms, the results drastically outperform the isolated efforts of a resource-constrained startup.
Assuming the startup chooses the right partner?
Well, yes. We are seeing tech transfer success rates of 85 to 95% when these structured programs are utilized. The 100-point scorecard, weighting microbial expertise heavily, combined with the specialized infrastructure of boutique CROs, provides a data-proven pathway that is vastly superior to a small biotech attempting to shoulder the crushing weight of GMP facility buildouts. The biological transfer has friction, yes, but the hybrid model is the only viable bridge across the valley of death for modern biomanufacturing. It is engineering triumphing over biological chaos.
And while it may be the only bridge we have, we shouldn't ignore the cracks in the concrete. I think we both agree unequivocally that ad hoc outsourcing, like simply throwing a strain over the wall to a CRO without a joint TEA in the first 4 weeks, is a recipe for complete disaster.
Oh, 100%.
And we certainly agree that if a small sponsor must outsource, they absolutely must seek out specialized boutique fermentation providers rather than monolithic generalists. But I maintain that as long as we treat living biological organisms like static assets to be transferred, we will be fighting an uphill battle against the very nature of the science.
It is a delicate balance, undoubtedly. Maximizing capital efficiency without sacrificing the fidelity of the biological process is sort of the defining tightrope of our era in biotech. And as we look ahead to the projected trends for 2026, particularly the integration of advanced AI-enabled DOE platforms and predictive digital twins that promise an additional 20 to 30% compression in development cycles, there is clearly much more to explore in how technology might further smooth this transition.
Indeed. It all comes back to that central reality. You can't just compile the code and run it on a new server.
The cell is the factory, and the factory is alive.
A living blueprint, constantly adapting. We will leave it to our listeners to weigh the economic imperatives against the biological realities, evaluate the frameworks, and draw their own conclusions on the optimal path forward for small biotechs. Thank you for joining us on The Debate.
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Ernala. Thank you for joining me. Until next time, keep engineering biology into reliable manufacturing.